I'd like to call on Eitan Wine next from Edmonton. He's the very hard-working treasurer of the Porto group and an expert in the microbiome as built up by his friend and colleague, Professor Dan Turner. No pressure, Eitan. Delighted to hear your talk.
Thank you so much for the invitation. I have to say that this is going to be a very hard act to follow, but I am talking about the microbiome, and everyone loves the microbiome, right? Okay, I'll get going. It's been a very difficult task that I've been given, and certainly, there's a lot of interest in the microbiome, but I am the first of 4 speakers that are going to give you a flavor of what some of the future will be, and I'll try to make it interesting, but given the short time, I'm only going to be able to touch on some of the few topics. These are my conflicts of interest, which are not relevant for this talk. So I'm going to start first by we all know what the microbiome is, but I'm actually going to try and define it. And I'll give some basic aspects that I think are important for us to understand as we move into this. I'm going to spend most of the time talking about 3 studies very briefly that I think are certainly very new. 2 of them were published over the last month. And they give us a bit of a flavor of where things are and where they might be. And I'll try and then give a bit of a glimpse into what this might look like in the future. So the microbiome was actually originally defined as the genetic material found within microbes, but that term has really loosened up over time, and this is what the NIH defines it as: the collection of all microbes, including bacteria, fungi, viruses, and their genes that naturally live on our body or inside us. And it really reflects this idea that this is an organ within us. And in many ways, in the environment, right, there is a microbiome that's related to human health, but certainly other microbiomes are of interest, the ocean water, the forest, etc. And they are now recognized as a very important part of the existence. This is taken from the APC in Cork, Ireland, and it really shows a lot of the interesting facts. A couple that I'll just point your attention to because they'll come up during the talk. So within our intestine, the microbial genes outnumber our host genes by a factor of 150. The other point is that we have many more viruses than we have bacteria, and that's going to be relevant when I talk about phages. And then the individual patterns of microbes, which is one of the biggest challenges. It's very difficult for us to define normal microbes, since every one of us here has at least one unique microbe that no one else has. So that's what we have to keep in mind. We also have to remember that there are many different factors that will impact the microbes, and they're listed over here. Some of these will be relevant to what we're talking about. I would argue that every single one of them is relevant to IBD. And when we talk about different omics, different factors, different exposures, the microbiome is likely a mediator of many of these, and the organs that are affected are listed over there just to give you a bit of a flavor. But because it's difficult to find microbes, the term dysbiosis has come up as something that I think we all have a general concept around, but is really a very problematic term. And I think that when we use that, we always have to put quotation marks or a bit of caution around that, because the best definition is something like a microbial imbalance, or having more of the bad guys and less of the good guys. But obviously that's a very loose definition because we don't really know how to define normal. But what is clear is that changes in microbes are associated with many diseases, and IBD is certainly central and probably one of the most important ones that we can discuss. So here's the other challenge. When we talk about the microbiome, and this study includes other omics as well, it really comes out with a lot of complexity and difficulty understanding. And I'd ask someone in the audience to please raise their hand if they're able to understand everything that appears over here, because I certainly can't. This is a leading study that was published a couple of years ago in Nature, and they looked at every omics that you can imagine, so the microbiome, the metabolome, the meta-transcriptome, metaproteome, etc., and tried to define these features with a large cohort of IBD patients. And it really is humbling to all of us, I think, how technology has advanced, but also the gap that it's created in our ability to understand this and to use it. And I'll give some examples that hopefully will bridge that gap, but we still have a huge way to go. And certainly, as most of us are clinicians, translating this into our care, I think, is a very important challenge. So I'm going to try and distill some of the major concepts before I talk about the studies that I've selected, just so we have a bit of a general idea of what this is all about. So certainly microbes are different in IBD. Now, we don't really know whether it's cause or effect in all cases. I would argue that we're seeing both of them, but that's an important thing to remember. Another important point is the host-microbe interaction. So microbes are not just living within our body, they are speaking to our cells, they're affecting us, they're also affected by us. And that's a major theme that will come up in some of the examples that I give you. The complexity of the microbes is measured in many ways, the many omics. Metagenomics is a major way of looking at that, which is looking at the microbial genetics. And then the interaction with the environment and environmental exposures is very central to that. And you'll notice that these three items are the exact topics that the next three speakers will be talking about. And I think that's an important message, and maybe why the selection for these topics was for the opening session, understanding the complete biome that we all live in and we're affected of. I should just mention quickly the hygiene hypothesis, which does link microbes to the development of IBD, the importance of looking at function of the microbes versus only composition. And then the last 3 points over here are really going to be exemplified in the next few slides that I'll give, so I'm not going to spend too much time at this point over here. So the 3 examples that I give were published, 2 of them over the last month, one a couple months earlier in Science, Cell, and Nature. And we're seeing more and more IBD studies in these journals. The reason for that, I think, is because microbes are identified as major interest to all conditions, and our biological tools for measuring them is really quite central. So to me, it really is humbling to see these great geniuses of the world who are now paying attention to IBD, and I would argue very specifically to pediatric IBD. The first study is going to be looking at microbial components that directly impact the immune response. The second one is looking at metabolites that these microbes produce, specifically bile acids. And then the third one will be introducing a very novel way of manipulating microbes. And then I'll just put a plug in for some of the work that we're doing, some of which will be presented here, where a lot of our focus is in the interaction between diet, microbes, and IBD. So this is the first study that I chose to discuss, and it was published in Science last month. And what was really interesting here was building on well-known observations. So if you look at dendritic cells as antigen-presenting cells, it's been known that they are able to take up bacterial extracts and introduce them to T cells, which will then promote the protective CD4 intraepithelial lymphocytes together with T regulatory cells and offer an immune regulatory dampening of immune response, which could potentially be used to treat IBD or certainly to prevent it. But the exact microbial factor was not discovered until this paper came out. And it really is, I won't go into too much detail, but a very elegant study where they did identify over here that the beta-hexosaminidase, which is actually a bacterial enzyme, is the factor that's able to directly stimulate by the antigen presentation those T cells to increase or reduce the Th17 response. They showed this in an animal model, and in the diagram over here, you can see the difference between the healthy state, where we do have the presence of the correct microbes and their metabolites, versus the disease state where they're absent and other microbes are producing a pro-inflammatory response that is not being regulated. Of course, there are many more details, but this is just the gist of the study. Also looking at the ability of microbes to suppress immunity is this study published in Nature. And what it looked at is the metabolites that are produced by bacteria. I think many of us know about bile acids, so they're produced by the liver, of course. Primary bile acids are then metabolized by bacteria into secondary bile acids. And some of these bile acids, such as glycolic acid that's shown over there, or 3-oxy-LCA, have been known to suppress, again, the pro-inflammatory immune response, and this paper was able to find the specific mechanism for that. Why is this important? Because once we know the mechanism, this host microbe interaction can potentially be translated into a drug, and that's the idea over here. So in a bit more detail, they first identified the specific bacteria that will stimulate this response. They then showed the exact bacterial enzymes which are doing this. They're converting that specific primary to secondary bile acid in different mouse models, and it was quite a complex study, and I had the privilege of, for gastroenterology, putting a summary of this in GastroDigest. So they were able to show that this is able to prevent the development of the disease, and furthermore, in humans, they showed that there's a certain profile of this enzyme that seems to be associated with the development of disease and severity, specifically in those individuals who have dysbiosis. So in other words, if you have the wrong bacteria that are not able to produce this metabolite, then you're at higher risk of developing IBD. And the third paper that I'll mention over here was published in Cell about a month ago. It's out of the Weizmann Institute. And unfortunately, I can't go into too much detail because it's a very complex and interesting study. They started this by looking at 4 different international cohorts of IBD patients and identifying that in all of them, Klebsiella pneumonia, which many of us know from other fields of medicine, seemed to be a recurring pathobiont that appeared again in patients that were developing severe disease. And that on its own is an interesting finding and quite important, but we have found other pathobionts in different studies, so I think that on its own was big news, but not the biggest news. But what they went on to do is find a novel way to actually eliminate those specific microbes. The way they did this was by using phages. So phages are viruses that attack or interact or infect bacteria, and they had to use a combination of 5 different phages in order to get stable suppression of these Klebsiella. They first were able to show that in vitro. They developed a model over here. They then tested this in animal models, and this is just the graphic abstract showing that if you infect with the bacteria, you get disease. If you infect with the bacteria together with the phages will lyse the bacteria, and you don't get disease. And what was really interesting was the human validation. So initially they did this in an artificial gut model, which is a very complex bioreactor model that's used. And they showed that it survived the gut and was able to move from the stomach model to the others. Although they did find that the pH probably eliminates these phages. They then gave human volunteers omeprazole to suppress their pH, and they found that they were able to get sustained activity of the phages and elimination of microbes in the model they were trying. They did not infect the humans with the bacteria, but certainly this opens up a possibility for the first 2 studies showing how microbes and their products can, in a way, improve our regulatory pathway. And this is an example of how manipulating microbes can potentially eliminate the pathobionts that are fueling disease. So this really fits into different approaches of altering microbes as a potential therapy, and this is an excellent review that includes a few different approaches. I discussed the bottom one, which is looking at the proteins and metabolites that are derived from bacteria, but I'd like to spend a couple minutes just on diet, just because that's something that I think in pediatrics we have a bit of an advantage. We know that diet works in Crohn's disease, but the mechanisms still aren't clear. And the whole idea is that if you have a certain diet that impacts your microbes and impacts disease, maybe we can do that reverse and try and correct this status. So the Crohn's disease exclusion diet will be mentioned further in this conference. I'm not going to go into too many details, but we did find associations with Johan van Limburger's work with the microbes in that setting. And tomorrow morning, Rotem will present the first data from our diatomics follow-up study on the Crohn's disease exclusion diet. We haven't done the omics part yet. We just have the clinical data, but I'm hoping that that will help identify microbial pathways and predictors that will be able to improve our understanding and treatments. In ulcerative colitis, this is quite big news. This is a study from India that's now in press and gut. And what they did there was use fecal microbial transplantation for 7 weeks, followed by an anti-inflammatory diet compared to a placebo arm. And they showed quite impressive findings, showing that manipulating the microbes in ulcerative colitis, and I think, Dan, what you presented shows a good rationale for this, may be a viable therapy down the road. Now, we're still waiting to hear some of the mechanisms involved, but this is certainly quite intriguing. And then one project that we've been working on through the Canadian Children's IBD Network is trying to see whether previous diets can impact your microbes and the gut environment and then predict your response to exclusive enteral nutrition specifically. This paper is currently under revisions for IBD, but what we did find through looking at dietary patterns in 100 patients who got exclusive enteral nutrition and we had food frequency questionnaires for, was quite complex interactions between your diet, the microbiota, and treatment. And some of these do suggest that the effect may indeed be through changes in the microbes, which will serve as markers, but possibly as guide for therapy as well. And finally, work from my lab. 2 of the people who have done this work are here today. So Heather, who was a postdoc in my lab and now has her own lab in Winnipeg, will be presenting this poster tomorrow. So I invite you to see it. And it relates to the fiber study that some of you may have heard of. Here we were trying to define specific pathways by which fiber might be inducing a pro-inflammatory response. And on the right, Michael, who's currently a postdoc in the lab, is looking at how short-chain fatty acids impact the host microbe interaction. So just to summarize, what might the future look like based on everything that I showed you? So I think We all know that microbes have something to do with IBD, but I think in the future we're going to be using microbes for prediction, and they really are a great way to understand the microenvironment in our gut. So they are not coming there to cause us damage. They are just adapting to the gut environment. So for example, if we have more aerotolerant bacteria, that probably means that there's more active inflammation, and that may end up being an excellent marker for disease. Using microbes or their products can certainly be a viable way to treat disease that we haven't done before, and then altering microbes as a way to change the biology of the gut, I think is certainly an attractive way to look at this as well. So I think the microbes will certainly be involved in many of the things that we're talking about in this conference today. With that, I'd like to end. Just before that, I'll thank all the people who did some of the work that I showed you. All of those underlined are here today at the meeting, so we have a nice representation of Edmonton. And I'll also mention again the masterclass that was mentioned earlier on. And I think we have a couple minutes for questions. Is that the plan, or is that not the plan? Okay, well, thank you very much.
Thank you, Eitan, for that beautiful review of the microbiome. So, of course, the microbiome in the internal environment reacts with very important cells in our gut, and that's the immune system. And we're absolutely delighted that Janneke Samson from the Erasmus in Rotterdam in the Netherlands is going to come today and to tell us what's hot in immunology with respect to IBD.
Thank you very much for the wonderful invitation. I've already had a wonderful day. The IBD Science course was fantastic. It's lovely being here, and the rest of the program also promises a lot. I have to agree with Richard's point of that it's so important to rely on communication and discussion to move forward. And I'd like to add to that what I have so much appreciated in the past few years is discussions with me as a basic scientist and clinicians to move forward. And I think that is something very special about pediatrics, and I think that is something that really should be cherished because that has helped me a lot in my basic science. So without further ado, I also have only 15 minutes to give you highlights of immunology. And this, of course, is an impossible task, but at the same time, it's nice because it really forces you to think, okay, what is truly, truly important in the next years to come? And what I did is I tried to combine results from literature in adult IBD, but tried to also relate that to pediatric IBD to see whether this also accounts for pediatrics. So it goes beyond saying that pediatric inflammatory bowel disease, just as adult inflammatory bowel disease, involves both innate and adaptive immune responses, and that these need to cooperate to really drive the disease. So what's very important to realize is that underneath this inflammation, both hyper as well as hypoimmune reactivity can lead to intestinal inflammation. So ultimately, this becomes a chronic disease, and even if you have temporary remission, you see that the disease flares. Now, this flaring comes from the adaptive immune system. This is because immune responses are made to provide you with long-term memory. This is the principle of vaccination, and especially CD4 T-cell memory responses have been implicated in the chronicity of IBD. So what are these CD4 T-cell responses? These are T-cells. In this case here, you see a naive T-cell, and that naive T-cell has a T-cell receptor that can recognize bacterial fragments from pathogens or microbiota that translocate into the intestine. Now, these are presented by antigen-presenting cells, in this case a dendritic cell, and that dendritic cell gives to this naive T-cell cues, co-stimulation as we call it, and cytokine signals. And this is needed because this dendritic cell is a good sensing cell and can then drive a particular program in that naive T cell to differentiate into a T helper cell that is specifically geared at eliminating the pathogen from which this peptide is derived. Okay, so there's a big different flavors of T helper cell responses. So this is, for example, a T helper 1 response. This program is really made to make interferon gamma and to eradicate extracellular pathogens. So there's more. There's the Th17 cell, in principle, this is really a protective type of cell that's very important for barrier function and antimicrobial peptides. But of course, we know that in ulcerative colitis, sometimes we have too much of this IL-17 producing cell. There's also regulatory cells like inhibitory function, IL-10 producing cells, or FOXP2 regulatory T cells. So these are about the different flavors of programs that there are once this naive cell differentiates. Key is that a proportion of these cells, once they've had their function and participated in inflammation, a proportion of these cells will remain. And once they remain, they can still be modulated. So that program in the cells is not fixed, and that is depicted here for Th17 cells, because we know that Th17 cells can reacquire an interferon type 1 type of program and then convey pathogenicity in immune-mediated inflammatory diseases. So these hybrid types of cells get multiple names, as we immunologists like to give our cells endless names. I'll be calling them Th1 star cells. That's because this is related to a particular function. What is key to realize is that these hybrid types of cells can also express multi-drug resistance genes and therefore be more resistant to therapy. So what is then so important about that memory response? What happens in memory is that these cells that have differentiated with their particular program reside in the lamina propria in the intestine, in these long stretches of tissue, quite inaccessible for all these treatments. And what happens is that these cells, when they re-encounter this antigen, so this peptide here in the T cell receptor, they can reactivate. And they do that very swiftly, but also have much less control from this antigen-presenting dendritic cell, because sufficient is only that peptide. In practice, this can happen in intestinal lamina propria, but there's also something called central memory. That means that these memory cells are also sitting in the mesenteric lymph nodes. Now, this is different from a primary T cell response, because as I told you here, there's much more control of what that T cell is going to do. So how does that work when you actually give the full picture of IBD? I realize this is a complex scheme, but what happens is that commensal bacteria are translocating and they go into the lamina propria. Here they are picked up by these antigen-presenting cells, and these antigen-presenting cells can be either hyperactivated or suboptimal in their function, or are participating in a network that is suboptimal in function. But these cells will migrate through the lymph, and then in the lymph node, these naive T cells will be primed. They will be primed, they will undergo that differentiation program, and then after a few rounds of proliferation, will move into the blood. When they go into the blood, they will respond to a chemokine gradient. Just means that there's factors that are calling these cells out to go back to the intestine. Now, these chemokines are, for example, CCL20, which is typically a chemokine that calls in Th17 cells. So it's not that just randomly all these cells will move into the lamina propria. The tissue really decides whether these cells are accessing the tissue because they make these gradients. Now, alpha-4 beta-7, the target of vedolizumab, is needed for these cells to exit the circulation, and the ligand of alpha-4 beta-7 is expressed on endothelial cells. So these T cells will then move out and go into the tissue. They've undergone that primary programming. They can then do their effector function, and that means they need to interact with all these innate cells to eradicate the pathogen. So once that is done, there's a part of these cells that are going to maintain there, and these are the memory T cells. Upon re-encounter of these commensal bacterial fragments, these memory T cells that are lying here or here in the lymph node are going to be reactivated. And this is what happens time and time again when disease is flaring. So what do we know? Well, we don't know a lot about these memory T cells in IBD. What do they respond to? What is their phenotype? We don't know how do these memory cells actually become activated in the lamina propria. What are the receptors they have for all these cues from antigen-presenting cells? And especially, we don't know how to monitor these memory T helper cell responses in individual patients. And as said before, if you want to move towards tailored therapy, you need to know a lot more about these responses. So a paper that came out, or actually two papers that were really crucial. One was by Chuck Elson's group, and the other, he's a collaborator on this, was on antibody responses to Lachnospiraceae flagellans. So Lachnospiraceae are commensals, and it's been established quite long ago already by Chuck Elson and Steph Targan that in health and in disease, we have antibodies to these commensals. That means we are seeing our commensals even if we're healthy. But in Crohn's disease, 60% of these patients have elevated IgG responses to multiple of these flagellants. And that really relates to the complicated course of the disease. So this is also true in pediatrics. We know that this even arises prior to disease development. But key is that until now, we didn't really know how these IgG responses related to CD4 memory responses. And to get an IgG response, the immune system needs to have CD4 T cells helping in order to get a productive IgG. So what Chuck Elson did, he used this novel type of assay that there is to really fish out these CD4 memory T cells from patients with elevated IgGs to flagellants. And they found that these cells had a particular phenotype, and produced interferon gamma or IL-17, and that there was a strong correlation between the number, so the frequency of these CD4 T cells and the anti-flagellant serum IgG response. So is this also true in pediatrics? Well, for that, we went to the PIBD SETQ cohort. This is a new cohort that was set up. If you want to know all the details, please see the presentation by Rens Kronberg in poster 069. It's a cohort that we run with multiple countries, multiple labs. What we do in Rotterdam is we collect a special cohort where we do a lot of biologic analysis. And so what we do is we include patients at time of diagnosis, and we have pediatric CD, pediatric UC, and patients suspected to have IBD but negative for the disease, and age-matched healthy controls. And we follow the patients longitudinally and have very precise clinical data. So what we did is we also measured with Chuck Elson whether in the same panel that he used, we could see reactivity in 94 pediatric patients of this cohort. And indeed, we found strong reactivity. And here what you see is the number of flagellants, and here the number of patients responding to that number. And then you see that there's a subgroup of patients that really responds to multiple of these flagellants. And these are mostly Crohn's disease patients. In fact, it's 41 of these Crohn's disease patients, and it's relatively little in UC. So that means that this reactivity is also occurring in pediatrics, which was known already before, but also in this cohort. So now the proof of the pudding was, and we just did a preliminary experiment here to see, can we also find these flagellin CD4 reactive T cells? And we can. So for us, this is crucial because it opens opportunities to study these pathogenic T cells in much more detail, understand their T cell reactivity, their phenotype, how they are activated, et cetera, et cetera. So why is it important for the clinic? Well, I think this is important if you look at literature, is that in all the animal models where these responses were studied, these were always animal models where there was an insufficient innate reactivity. So toll-like receptor 4 deficiency, that made these mice more susceptible to an infectious trigger, and then there was bystander commensal translocation. Now, when that happened, the defective innate immune response could not eliminate these bacteria, and that caused a strong T helper 1 memory response. And that long-lived memory response, even after this initial trigger was gone, was maintained and caused reactivation of this IgG response in these mice, similar to what we see in the patients. So the hypothesis is that also in these patients, there might be an issue had insufficient innate immune reactivity underlying this whole response. There's evidence for this that NOD polymorphisms associate, NF-kappa B polymorphisms associate, but these are all little pieces of the puzzle, and I think that we need to look into that a lot more in the future, because I think manipulating bacteria and host immune system may be good for these patients. So last few minutes on this memory response. I'd like to say that ustekinumab, of course, is a very important drug now in adult IBD and becoming more and more used in children. But there's new drugs coming up, especially the selective IL-23 targeting. Now, what is nice about this is that IL-12 is really important for this differentiation of the naive T cell, but actually doesn't express an IL-23 receptor, whereas IL-23 receptor is much more expressed by memory T cells, and therefore I'm curious about these results and would love to see more immunology in these trials that are being done to see how this selective regulation of memory is going to be affecting the disease. So, as said before, we need to be able to much better type this in individual patients. We and many others have been working hard on identifying these pathogenic T cells. Currently, we've been successful in doing that by doing a complex flow cytometry protocol. What I'd like to tell you is that indeed you can visualize in individual patients these Th1 star cells. And so this is in pediatrics, also in the PIBD SEDQ cohort. So I think that enumeration in patients in time is possible. The question is, should you be using that in the clinic? I think it's probably too complex, so in that sense, I agree with you. But there are many other types of methods, sampling, that are easier, that may definitely relate with these types of responses. And so what I'm showing you here is these new proteomics types of assays that there are. For example, the O-link technology. I saw that George was also going to be speaking about this. These are highly sensitive, specific, and high throughput. And so here you see interferon gamma and IL-17A, and you can clearly appreciate in pediatric CD of PIBD SEDQ, there's a large variation in the degree, so the concentration of interferon gamma in plasma at time of diagnosis. And you see that in general, Crohn's disease is higher than ulcerative colitis, but there's certainly patients that are almost similar. So this really calls for looking much more in detail to see how can these profiles of many immune proteins, not only interferon gamma and IL-17A, but also, for example, all these chemokines, help us to make patterns in different patient groups and see whether these patients have different types of disease courses and disease severity. So with that, I think I have tried to explain you what immunology is about in a nutshell and what we are looking forward to. I would like to stress that I'm in the luxurious position of having great collaborations to do all this work. So Hankje Escher and Lizzie de Ridder are here, and many of our group are actually here. From the clinic. My lab, we do everything fresh, so that means a lot of night work to do all our analysis, and I have a superb team to do that. We have great collaborations in bioinformaticians. I'd like to thank Frank for in this initiative of PIBD SEDQ, and we're working closely together with Work Package 7, which is Nick Croft and Chronis Chemos. I'd like to thank you for your attention.
Thank you, Jannica. That was absolutely brilliant. I really enjoyed that. Now, if you don't know this next guy, this is Professor Ho Mulig. He's from Oxford. I think, personally, he's going to appear in an episode of Inspector Morse or something like that very soon. He has a brain the size of a small Scottish island. And if you ask him any question about genetics, he's going to know the answer, but he's going to give us a snapshot into his mind for the next 15 minutes.
Thank you very much, Richard and David, for setting up this meeting, inviting me. And yeah, I want to discuss what's hot in IBD etiology, the genome. Obviously, this all depends on definitions. And if you're very, very strict, there are only two things that are probably hot. Your cycler, your PCR cycler, and then the chips that you use sort of in your server farm to analyze all this data. Now, what we like to do is we all know there's a genetic component to IBD. This comes from data 30 years ago on twin studies. There is about 20 years of GWAS and GWAS-like association studies. And what we really like to understand, what are the causal variants? What are the functional mechanisms that are associated? And what we have seen over the last few years is a dramatic change in technologies from big sequencing factories towards point-of-care technologies. It becomes cheaper. £1,000 was the target for a genome until 2 years ago. Now it's £200 or $200 sort of even. There are now predictive tests. We really want to learn what this tells us about our patients. Is there anything that we can learn to change our treatments? What we have seen over the last several years is a dramatic increase in functional genomics, linking these variants to epigenetics, to transcription, to isoforms, and to immunology. Now, I want to give a few vignettes that have data that have emerged since PIBD 2019. One thing is scale on everything. So several years ago, it was about 1,000 patients. Now we are hundreds of thousands. It's a recent paper from the International IBD Genetics Consortium with contribution of several pediatric gastroenterologists. And these are 30,000 patients who have been sequenced with exome and genome sequencing. And the number of genetic variants is increasing. And what this tells us is we are now sort of reaching this area where it's not the common, but rarer variants. And in this field of rarer variants, we see that there is an overlap between the monogenic variants we have seen and these polygenic variants, as indicated by variants, for instance, in REL-A or IL-10RA. And this suggests that there is some overlap in the genetic composition and susceptibility. Second, one of the huge problems that genetic has is to explain a practical purpose. And this is despite hundreds of variants now available and credibly associated. Not one of the common variants has made it into clinical practice. I would argue zero. If you look at the guidelines, do you look genetics? Do you look for NOT2, ATG16L1, IRGM? All these variants, no. They are very, very clear. We have brilliant evidence. It tells us something about the mechanisms, but it's currently clinically not relevant. Another large part of these variants is not in the protein coding parts. It's intronic or intergenics. And we need to understand sort of which genes these variants interfere with. And there are now sort of several genetic association studies that look for the regulatory phenotypes where these variants intersect. Interestingly, several of the IBD variants are in loci that are shared with other immune-mediated diseases. And we are just about to understand that not every locus is linked to the same gene, and there is quite a bit of work to be done to understand how this affects the epigenetic landscape, how these genetic and epigenetic loci intersect, how this affects transcription, and what is the practical consequences, not just for a body, but for individual cells. This will be informative, given that we have now sort of a revolution in transcriptomic technologies towards a single cell level. The Human Cell Atlas tries to understand the cellular landscape. There are studies in pediatric inflammatory bowel disease in adult IBD, on healthy controls, how the cellular landscape develops over time, and what is the change in inflammatory bowel disease. And these can have therapeutic consequences in the future, because there are now emerging data, for instance, from Seattle and from the Boston Children's Hospital to suggest that the cellular composition on a single cell level can be associated with disease severity and the therapeutic progression, for instance, towards biologics. Now, we typically think about 16 to 19,000 genes that our genome encodes. But actually, there is quite a bit of detail in there, because we have isoforms. And typically, most people think about sort of only the canonical variant. There is now emerging data that in the IBD pathophysiology, there is a dysregulation of splice variants as well, and this will open up dramatically the field we have to study, since several splice isoforms can be encoded in the same genetic region. There are now variants that probably have therapeutic impact, and there's this field of emerging pharmacogenomics. And the PANS consortium, led by Tariq Ahmed, has identified variants that are associated with toxicity, but similarly with response to therapy, with development of immunogenicity towards biologics. And Jack Stansaki and Nicola Terit have further investigated this data set. And there is some data to suggest that HLA haplotypes might predispose to this immunogenicity, and there could be differential effects between, for instance, adalimumab and infliximab, relevant information. In addition to these polygenic forms of inflammatory bowel disease, this rare field, the small ones, the underdogs of monogenic inflammatory bowel disease, have moved over the last several years into clinical practice. And I'm very grateful to our colleagues and friends of the Porto group that we were able to set up a practical and pragmatic position to guide the transition of genomics into clinical practice. And this means that in several healthcare systems, there is now genomics available to patients with a suspicion for monogenic forms of inflammatory bowel disease. The value of this has been shown, for instance, by a study by Alexio Muis in Toronto, showing that if you do trio sequencing in a large cohort of patients with pediatric inflammatory bowel disease, it reaches the low percentage level of possible variants that should be followed up and that have potentially a therapeutic consequence. But this field is not saturated, because since 2019, we have seen at least 20 new monogenic conditions, and there are still several years, a few more to come. And this is really a community work. This is possible because colleagues in Munich and in Toronto, in Boston, in Vienna, in Philadelphia, and at the NIH in Paris have shared forces and worked towards identifying genetic causes of monogenic inflammatory bowel disease. We were able to set up a taxonomy of these diseases, and that allows us now to study key shared mechanisms that affect multiple layers of the immune system. This immune system that Janneke has mentioned, and this has potential therapeutic consequences, because if we see new genes that are in the same functional and pathophysiological class, it's more likely that they will respond to similar treatment options. Now, the aim was that genetics informs treatment. And there is now sort of emerging evidence from the polygenic as well as from monogenic studies that either-Mendelian randomization approaches in polygenic disease or case studies, careful case studies, can inform about treatment options. But the ultimate goal is a cure. And closest to this are gene therapy approaches in these forms of rare monogenic forms of IBD. And this has been now shown in several approaches, initially in retroviral studies, now in more safer lentiviral studies, that you can set the genetics, introduce, correct the disease copy, and this has led, for instance, to the resolution of intestinal inflammation and severe Crohn's disease-like intestinal inflammation in patients with chronic granulomatous disease or in Wiskott-Aldrich disease. Now, I want to finish that there is a moving target. I mentioned already the reduction in price. This will drive more and more genetic studies, but on time as well. And for instance, these new technologies that are available reduce the time from sending a sample towards the genetic results dramatically. I think we all know the time when we waited for a year, 1.5, 2, for research results about genomic testing. With this clinical genomics, hopefully, is now several weeks. But there are now studies available who have shown that ultra-rapid sequencing is possible. And in the intensive care setting, the lowest time from sending a sample towards sort of the results, it has recently been published in the New England Journal, and it is 7 hours and 18 minutes. So within one shift on the intensive care setting, you can have a diagnosis. Admittedly, that is rarely needed in pediatric inflammatory bowel disease, but I'm pretty sure we have all seen some patients who sort of progress towards colectomy, where a fast exclusion of a monogenic defect would be very helpful. And I'm pretty sure we will see these technologies in clinical practice soon. Summarizing, since last PIBD 2019, genomics has arrived as standard of care in clinical practice. There are shared and distinct mechanisms between monogenic and polygenic forms of inflammatory bowel disease, and this does inform treatment options, including the most dramatic form of gene correction, gene therapy, in some forms of monogenic inflammatory bowel disease. Thank you.
Thank you very much indeed, Holm. So the last of our 4 speakers is Charlie Lees, our colleague here from Edinburgh, adult gastroenterologist at the Western General Hospital and a scientist in the University of Edinburgh. And Charlie's passion, for which he is an exceptionally effective communicator, is the exposome, the environmental influences. So we're really looking forward to this, Charlie.
Thanks very much, Richard. Thank you very much, David. Thank you for organising this amazing meeting and for bringing all of you wonderful people to Edinburgh. So welcome to our great city. So I'm going to talk now about the exposome in IBD, by which we mean the environment and environmental triggers in IBD. And I'm going to talk a bit about the role in disease etiology and a bit about the role in disease course. And I'm going to try and give a few practical takeaways through this as well, whilst bringing together some of the themes that we have heard already this evening. So the gut really is a central part for much of human health and disease. And within that, it mediates a lot of the environment through the gut microbiome, as we have heard already. And this really is true more than anything for gastrointestinal health and for inflammatory bowel disease as well. Conditions that we know are getting more and more common, where there is dysregulated immune response and altered microbiome, and clear environmental and genetic drivers, with a series of important clinical problems, many unanswered questions, and an urgent unmet need that we share as both adult and pediatric gastroenterologists. And within all of this, within all these unmet needs, we really need to keep an eye on the future that we and our patients demand, which is a roadmap leading to the prevention of Crohn's disease and ulcerative colitis. And when we think about that, it's really important that we get a true understanding of how the environment and the rapid environmental shifts over the last century have been influencing the development of inflammatory bowel disease, and that will help us lead further towards prevention strategies over time. So who gets inflammatory bowel disease and why? Richard and David and Dan have shown us already some of the historical perspectives from Scotland to North America, looking at what's happened with the environmental and epidemiological shifts over a relatively short period of human history. And within that, we've seen these really remarkable trends, and trends that are going to continue over time. Now, it's important for us to recognize that we've known about inflammatory bowel disease in the historical and the ancient literature for at least hundreds, if not thousands of years. And when we come to think about environment, it means that we cannot and must not blame patients for falling into a fast food trap or something else in their environment that has led to their development of disease. We know causality is much, much more complex and nuanced than that. Nonetheless, we see that as the world has changed, the incidence and subsequently the prevalence of Crohn's disease and ulcerative colitis has increased dramatically. And this is not going away. It's only shifting further and to different parts of the world, as I'll come back to a bit later on. Gil Kaplan has very neatly described the 4 phases of IBD epidemiology. And we here in the United Kingdom, across Europe, in North America, and much of the westernized world are now in this 3rd phase of compounding prevalence. Gareth Jones here in Edinburgh has put together a beautiful study looking at the epidemiology of adult and pediatric IBD in Scotland, and in particular in Lothian, this region where we are that consists Edinburgh, consisting of 900,000 people. And we know that from the middle of 2018, the point prevalence was 0.8%. We see the age at diagnosis, mostly in young people, these people who are getting older. And because incidence is stable, but because thankfully mortality is not dramatically increased, we are seeing this compounding prevalence. So already we're half the way along this graph, showing that we will be hitting 1% prevalence in Scotland by 2028. So what's going on? Our environment is shifting rapidly. With the odd exception, like cotton-top tamarins, we don't see colitis in primates. We probably saw it occasionally in ancient man, but this really is an illness of the last century and the second half of the last century. And there have been some very dramatic shifts. This looks at the world's population and those in urban versus rural areas. The urbanization of our population seems to be the key driving environmental change, following which you see an increase in adoption of multiple different technologies that include flushing toilets, running water, refrigeration, microwaves, home air conditioning, many of which may be important clues as to what's going on. Within a similar time frame, we have seen a dramatic decrease in infectious illnesses through vaccination and through widespread antibiotic use, which we know appears to be an important factor in shaping the early microbiome. We have also thankfully seen a huge shift in sanitation, but it is worth pausing to note that this is very different in many parts of the world. And if you look in the bottom to sub-Saharan Africa and low-income countries here in this Our World in Data chart, you can see unimproved or open defecation still very prominent. And this is something for us to think about globally, particularly in these regions where diarrheal illness remains a major cause of childhood mortality. So let's start to bring these factors together within inflammatory bowel disease. We've heard beautifully from Holm about the genetics, and from Eitan earlier about the microbiome, and of course from Jeanette how these influence what's going on with our immune system. The environmental factors have been listed here. There are many. Diet is probably one of the key factors, and I'm going to spend a bit of time talking about that today. But there are many other interesting aspects, including hygiene, dietary additives, geography, travel, smoking, perhaps sunlight, vitamin D, stress, industrial pollution, and other contaminants. But if we cut right forwards to this IBD consensus statement published in Lancet GastroHep earlier this year, we can actually start to distill this down into practical takeaways. What are the modifiable environmental factors that patients who already have inflammatory bowel disease should be advised more generally? And it's quite general, but there are some things that we know. Avoid smoking cigarettes, good for health anyway. Adopt diets that have proven to work best in clinical trials. Well, that perhaps is more for the future than it is for now, and I know that Kostas is going to talk a bit about that tomorrow. Light consumption of alcohol does not make IBD worse. Depression and anxiety may make IBD worse. Incorporate regular physical activity into your routine, maintain normal body weight and muscle mass, and obesity may reduce the effectiveness of drugs and increase the risk of complications post-operatively. Also sensible to avoid the regular use of high-dose non-steroidals, although there is some controversy in the literature about low-dose or at least sporadic non-steroidal use, and I think we're more relaxed about that than we were previously. And then very helpfully, there is also some thought about what this might do for children of people with inflammatory bowel disease. So if you're a parent with inflammatory bowel disease or a would-be parent, what do you want to think about in terms of stopping your child developing inflammatory bowel disease? Well, the first thing to say to any parent thinking about having children is go for it, because the chances that that child has inflammatory bowel disease remains very low. But nonetheless, many parents ask, how do I decrease the risk of IBD in my child? And the advice that's within this consensus statement is to encourage breastfeeding where possible, avoid unnecessary antibiotics in early life, and avoid secondhand cigarette exposure. These are all pretty healthy ways to bring up a child anyway. I would add to this list, consider having a dog as a pet and to limit the consumption of processed and ultra-processed foods. It's interesting that one of the three things they talk about is to encourage breastfeeding where possible, because David's very nice recent study with Christopher Burgess, looking at 2 million children from the SMR02 dataset across Scotland, born between 1981 and 2017, with 1,721 incident cases of pediatric IBD within this cohort, demonstrated that, in fact, feeding type of infants did not appear to have an influence on subsequent development of inflammatory bowel disease. The dog bit comes from recent data from the GEM study that was presented by Will Turpin at the DDW meeting in San Diego this year, where they demonstrated a lower risk of developing Crohn's disease with growing up with a dog between the ages of 2 to 4. That's the child growing up with a dog between the child's ages of 2 to 4, not the specific ages of the dog, and having a large family size of over 3 individuals in the first year of life. There was some association with altered gut permeability, but this was not consistent throughout, and no link was found between dog ownership and cow-protectin microbiome composition and diversity. So this is a curiosity. It didn't extend to pet cats, interestingly. Nonetheless, I would argue that, as with the other advice, having a dog in life is great for company. So let's move to think about diet in a little bit more detail. This is self-reported dietary choices of Brits in December 2021. This is what British people told this survey that they were eating. Were they meat eaters, flexitarians, pescatarians, vegetarians, plant-based vegan, or none of these? And you can see how it shifts by age. Now, those of you new to Scotland may observe some interesting eating behaviours whilst you are here, and some curiosities to our Scottish diet. It is rather sad that in some parts of the United Kingdom population, some people have up to 80% of their daily calories coming from ultra-processed foods, and this is a significant issue. When we start to think about the influence of diet on IBD, we are in large part thinking about the interaction between diet and the microbiome. And this needs a little bit of thought just to put our ideas and the data into context. This is the microbiome over life, and you can see that the weaning and life stages of diet are shown on the bottom. So you go from breastfeeding to weaning to solid foods to healthy diet, the impact of high protein diet, healthy diet, and in older age. And you can see the impact on diversity and some bacterial families. I want to bring your attention to in the middle. You can see where it rapidly squiggles up and down in that 24-hour period. So there is big shifts that happen. So you have to be quite careful about how we study the microbiome. We saw this from this beautiful study by David et al. In Nature almost 10 years ago now, where healthy people were put onto a plant-based diet, washed out and then put onto an animal-based diet. You saw the shifts in dietary fiber on the top, fat intake on the bottom, and then you saw, particularly when they went from the plant-based onto the animal-based diet, a significant reduction in the diversity of the microbiome within a couple of days, showing that some of these shifts happen very, very rapidly indeed. But what about in inflammatory bowel disease? We're starting to accumulate some very clear signals of association between some dietary types and inflammatory bowel disease. You can look at the risks in terms of omegas and PUFAs. You can look at fiber. You can look in Crohn's disease, as I'll come to, at ultra-processed foods, and ulcerative colitis, probably animal protein, and perhaps sweet beverages too. So let's have a think about a couple of these things, thinking about high-fat diet and also thinking about emulsifiers. I really like this quite old study now looking at high-fat diet, because what it does is it brings together, albeit in an animal model, on the left an environmental factor, the diet, which is a milk-derived saturated fat diet. In the middle, a host factor, and I think the metabolome will prove to be one of the most critical omes moving forward, thinking about bile acids in particular. In the microbiome, you see this growth of Bilophila wadsworthia, and then this only leads to colitis when you have a genetic variant, in this case, a genetically susceptible mouse. Emulsifiers have had a lot of interest in the last recent years. Emulsifiers are the things that make our food look and taste and feel nice. On the right-hand side, this is mayonnaise made without emulsifiers. When you make it with emulsifiers, it starts to look nice and tastes very much better. And this study by Bernard Chassing in Nature from a few years ago now demonstrated many factors about emulsifiers, but in particular, it showed that quantities of carboxymethylcellulose and polysol by 80 that the FDA considered to be safe and generally regarded as safe were associated with increased colitis in animal models. And Kevin Whelan and James Lindsay and others in London are now running the ADAPT study, where they're taking 150 people with Crohn's disease and then resupplementing onto a baseline diet snacks that are either containing or not containing emulsifiers. And I think this is a very interesting study indeed to see whether or not this gives us a modifiable factor for patients moving forwards. What about the data for ultra-processed foods? This comes from the PURE study. The PURE study is the prospective urban and rural epidemiological study. It's a big study. This paper was published in the BMJ. Was this study set up to look at Crohn's disease and ulcerative colitis? Was it? Heck. They were interested in looking at cancer and cardiovascular disease and multiple other factors. But some sharp minds got hold of the data set. They've reanalyzed it to look for Crohn's disease. In this cohort, there were 34 incident cases, and ulcerative colitis, there were 165. So it's important to bear these things in mind, even though this is methodologically sound and it's big numbers. It's not specifically designed to look at inflammatory bowel disease. What they did find was higher ultra-processed food intake associated with a higher risk of inflammatory bowel disease. That's the headline. But importantly, in the conclusions, a possibility also remains of residual confounding owing to unmeasured or unknown confounders. And indeed, when you look at the fully adjusted models, the significance really starts to creep away. This is really important to bear in mind. You need to look deeper than the headline figures. So that's ultra-processed foods. This is diet, and this is fiber. This is from Ashwin Ashkanathian's study from gastroenterology, from the Nurses' Health study. Similar, very large numbers, Quite a few incident cases. Bear in mind that the enrollment was between the ages of 30 and 55 years of age, and within this, you see an association with Crohn's disease, not ulcerative colitis, in terms of a protective effect of high amounts of fiber in the diet. But when you start to adjust the model, it goes away. And this is only a very partially adjusted model. So it's important clues and it makes sense. But we really need to test our assumptions in these studies. A brilliant study published in Cell this year on the left, nothing to do with IBD, compared high fiber diet with fermented foods diet. And the fermented foods diet was the diet that had a much better anti-inflammation impact than the diet with high fiber, really going against some of those assumptions. And then on the right is another study published in Cell this year looking at sweeteners, non-sugar sweeteners, and showing that they make a significant abnormality or at least dysbiosis in the microbiome. Test your assumptions. We need to think about this very carefully. It's very easy to fall into the lazy traps to say IBD is getting more common around the world because people are eating a different diet. Patients get very upset if we tell them that diet is the cause of their IBD. Nonetheless, we're getting better at doing this. The DINE-CD study gives us some clues as to how we can study the impact of diet in Crohn's disease, showing specific carbohydrate diet and Mediterranean-style diet. I tell my patients that a Mediterranean-style diet is probably the simplest thing to take. Moving towards the last bit here now, just thinking about the environment and the environment from a slightly different perspective, and that is when we study the environment as well as the microbiome, we have to be really impactful of the powerful effect of both inflammation, medicines, but also on people's behaviors when they're unwell. So we know that for the microbiome, what's cause and effect. It's also true for diet. So the GEM study is great because it looks at before diagnosis. We started to think maybe some of the dietary and environmental factors that are associated with triggering IBD onset in the first place may also be the same as triggering disease flare. And if we looked at patients in remission, we might be able to get a better understanding of what's happening leading to disease flare. So the PREDICT study that we've been running for a number of years now is set out to find which aspects are associated with and predict flare in Crohn's disease and ulcerative colitis, looking at diet, environment, gene variation in the microbiome. And this is done to test, or at least try to answer the questions that patients ask us. What should I eat? What should I do to stay well? And so to do this, we've recruited 2,500 people in clinical remission. We've done a deep dive into the microbiome, into calprotectin, into environmental surveys, dietary surveys too. We've followed most of these people for out to 4 years now, so we can accumulate soft and hard flares and compare the baseline variables in those two groups. We're primarily looking at dietary factors based from the literature when we set this study up: animal protein intake, dietary fiber, PUVAs, emulsifiers, and bacterial gene count. With broad inclusion criteria, this is a pediatric and adult study. We've gone from the age of 6 up. We've got 2,629 patients in the study. The study closed to recruitment in March 2020 at the time when the pandemic hit. This is from 49 sites across England, Wales, and Scotland. We've got a deep dive into a number of questionnaires at baseline, looking at a variety of different environmental factors as well as quality of life, and some other important metrics, as you can see here, including grit, resilience, sleep, stressful life events, exercise, etc. But the diet's the most important bit of this for us. We've got dietary surveys based on food frequency questionnaires. We've got, in a subgroup, 4-day weighed food diaries with stool for calprotectin and microbiome collected on day 4. We've got 2,600 patients, Crohn's disease and ulcerative colitis, and it's across all different age groups, as you can see. The dietary data is completely allocated now from the FFQs. Interestingly, a couple of tidbits. You can see 3% vegetarians, which is relatively low, and a huge array of other really interesting, impactful data. We've asked patients their perception of flares. You can see dietary changes, stress and alcohol, all these environmental factors are key in patients' minds. But really what's important is how do these baseline variables impact on our recorded flare rates and progression rates over time, shown by a participant on the left with negative calpros throughout, participant on the right where the calpro is going up. And we'll be able to share those data with you very soon. We're just cleaning up the databases now to do this primary analysis. So where are we before we go for a drink and dinner in Scotland, where Crohn's disease was first described, where we are in a world, really a world that is going to have to grapple with these issues that we've got. And I really think it is our job, all of us, to help the global IBD community here. I was in Egypt last month at the first Pan-Arab Crohn's and Colitis Association meeting, and I made a new friend there who's Suleiman Hussein. He is a surgeon who did some of his training in the UK. He went back with his wife 10 years ago to Khartoum, where he, from what I can tell, is now single-handedly managing the inflammatory bowel disease that they're seeing there, and they're seeing a lot, and it's similar to ours. Khartoum is one of the fastest-growing cities in the world. It now is home to 5.5 million people, and they are seeing more and more inflammatory bowel disease develop. This is where the real environmental shift is influencing what's happening, and perhaps this is where some of the clues are to what causes IBD and what we can do to prevent and cure it. Thank you very much for your attention.
Charlie, thank you. Thank you very much, Charlie, and thanks to all of the speakers. Thanks for all of you for staying through the whole day. It's been absolutely fantastic start. As your reward, the welcome reception is now open downstairs. Thank you very much.